Good morning, how are you? Today, inshallah, we will start with chapter 13, the RNA. Let's start with the RNA. Let's study the RNA structure, the difference between the RNA and the DNA, and the function of RNA, and finally the types of RNA. Let's start with the first one. How does RNA differ from DNA? Let's think. What's the difference between the RNA and DNA? Is there any differences between the DNA and RNA? What do you think? The answer is there are three important differences between the RNA and DNA. The first one, that the sugar that is found in the RNA is called ribose sugar, while the sugar that is found in the DNA is called deoxyribose sugar. Okay. The second one, the RNA is single strand, while the DNA is double strand. And finally, the third one, the RNA contain nucleotides called uracil instead of thymine that is found in the DNA. Let's start with the rule of the rule of RNA. Genes contain coded DNA instructions that tell the cells how to build the protein. So the DNA have the codes for protein building. The first step in the decoding, we need to copy the DNA into RNA. So we need to copy the DNA in the form of RNA. RNA is a nucleic acid that consists of long chain of nucleotides, but it's just one strand. RNA that uses the base sequence copied from DNA to direct the production of protein. Each nucleotide in both DNA and RNA is made up of 5 carbon sugar, phosphate group, and nitrogenous bases. The difference between the sugar that is found in the DNA is deoxyribose, while the RNA is ribose. Also, the RNA is single strand, while the RNA contains uracil instead of thymine. All of these differences between the DNA and RNA, please make sure that you understand and save all of the differences between the RNA and DNA. Also, the role played by the DNA and RNA are similar to the master plans and blueprint used by the builder. You have the blueprint, which is the DNA, and you will take copies from the blueprint or the master plan by the photocopier. The photocopies here are called RNA. Then you will use these copies to produce the protein. So the cell uses the DNA, which is the master plan, to prepare RNA, which is called the blueprint. The DNA molecule stays safely in the cell's nucleus, while RNA molecules go to the protein building sites in the cytoplasm, which is the ribosome. So the DNA will stay safe in the nucleus, while the RNA will go to the cytoplasm and to the ribosome to build the protein. You can think of RNA molecule as a disposable copy of a segment of DNA. So once we use this RNA to form or to build the protein, then we will dispose the RNA copy. So RNA have many functions, but most RNA molecules are involved in the protein synthesis only. So the main important role for the RNA to build the proteins. Also, the RNA controls the assembly of amino acids into proteins. Each type of RNA molecule specializes in a different aspect of this job. Let's now take a look on the types of the RNA. We have mainly 3 types of RNA. The first one, which is mRNA, which is called the messenger RNA. Then we have the ribosomal RNA or rRNA, and finally we have the transfer RNA, which we call it tRNA. Again, we have the 3 types: mRNA, rRNA, and tRNA. Each one of them have a different structure, different function. Let's start with the messenger RNA. Most genes contain instructions for assembling amino acids into proteins. The RNA molecules that carry copies of these instructions are known as a messenger RNA. They carry information from the DNA to another parts of the cell. So all of the information that's copied from the DNA is copied at the form of mRNA or messenger RNA, and this information will go to another place to transcribed into the proteins. Ribosomal RNA, which is the second type, proteins are assembled on ribosomes, small organelles composed of 2 subunits. So as you can see, this is the ribosomal RNA, contains 2 subunits. This is first subunit and this is the second subunit. The ribosome subunits are made up of several ribosomal RNA, which is made up of 80 different protein. We call it ATS, which is made up of 2 subunits, 50S and 30S. The third type, which is transfer RNA. When a protein is built, a transfer RNA molecule transfers each amino acid to the ribosomes as it is specified by the coded messages in the mRNA. So the transfer RNA will carry the amino acids needed to the code of mRNA. Let's start with the RNA synthesis. How does the cell make RNA? In the transcription process, the making of RNA, segment of DNA serves as a template to produce complementary RNA molecule. So transcription using the DNA as a template to build up the RNA molecule. Let's start with the transcription. The base sequence of the transcribed RNA complement the base sequence of the template DNA. So we have the template DNA. The code in the template DNA is, as an example, G, C, T, T, A. This is DNA template, okay? If you want to find the RNA which is complementary to this segment, this is the RNA, we will find against or the complement sequence for the GCTTA is C, G, A, and the A will face U, uracil in the RNA instead of T, okay? So this is the complementary sequence of this DNA sequence, and this complementary sequence is found in the form of RNA. Done? Is that clear? Okay. In prokaryotes, RNA synthesis and protein synthesis take place in the cytoplasm, while in eukaryotes, the RNA production will be in the nucleus, then it's moved to the cytoplasm and the ribosome to build up the proteins, okay? So let's start with the transcription in the eukaryotic cells. Transcription requires an enzyme known as RNA polymerase. So we have an enzyme here called RNA polymerase, which will start the transcription of RNA that is similar to the DNA polymerase. So RNA polymerase have a similar sequence or similar structure for the DNA polymerase and similar function. Look at the original DNA sequence and the forming RNA sequence. As you can see, we have here the thymine and the uracil. You will face here the thymine instead of uracil that is found in the DNA. RNA polymerase bind to the DNA during transcription and separate DNA strand. So it will separate the DNA strand. Then RNA polymerase uses one strand of DNA as a template from which to assemble nucleotides into a complementary strands of RNA. Then we have a promoters. The RNA polymerase binds only to the promoters, so again, RNA polymerase binds only to the promoters or to promoters regions of DNA that have a specific base sequence. So without the promoters, the RNA polymerase will not bind to the DNA. What is the promoter again? The promoter is a specific sequence on the DNA. Promoters are signals in the DNA molecules that show RNA polymerase exactly where to begin making the RNA. So without this signal, without this promoters, the RNA polymerase will not bind to the right sequence, okay? Similar signals in the DNA causes transcription to stop when a new RNA molecule is completed. So we have a different signals to stop the transcription, okay? After the copying of the mRNA or the RNA, we need to edit the RNA because the RNA or the newly formed RNA is composed of two units, introns and exons. Okay, these two parts of the RNA have a different sequence, and we have to know that the introns are not coded for the proteins, while the exons are coded for the proteins. So in this process, we need to remove or splice out the introns, and the exons must remain in the formed or the RNA that is formed after the editing. Okay, So we don't have a complete answer to why the cells use energy to make a large RNA molecule and then it will cut it to form the adult mRNA or RNA molecule. Again, transcendent exons may also play a role in the evolution, make it possible for very small changes in DNA sequence to have a dramatic effect on how genes affect cellular functions. After the RNA is edited, then the transcription process is finished. Then we will move to another process called transformation, and that process, inshallah, we will take it in the next lecture. Thank you and have a nice day.